So greetings, everyone. My name is Koryanko. I'm the interim curator here for the Draper Naturalist Museum at the Buffalo Bill Center of the West. And first and foremost, I just want to thank you all for taking time out of your busy schedules to come and hear an incredible and a very important story that really has its roots here in Wyoming. We are very pleased and honored to welcome you tonight for a special feature on the recovery, the conservation, and the cloning of the black-footed ferret, possibly one of the greatest conservation success stories in modern history. But we also want to recognize our partners, our panelists, and our special guests that are here tonight. Pete Gober, Kimberly Frazier, and Robyn Bortner from the U.S. Fish and Wildlife Service, Zack Walker, Andrew Gigley from the Wyoming Game and Fish Department, Dennie Hammer, where this story all began, Dr. Oliver Ryder from the San Diego Zoo Wildlife Alliance and the Frozen Zoo, and Ryan Phelan from Revive and Restore. Moderating the session tonight is Dr. Lenox Baker, owner of the Pitchfork Ranch and the place where this story really continues. So without further ado, and out of respect for all of our panelists' time, please welcome Dr. Lenox Baker.
We should be able. Can everybody hear me all right? Good. And I do welcome you all here. Talking to Pete earlier tonight, he informed me of a quote he heard once I thought was very appropriate for the beginning of this. And this is a quote from Charles Darwin in 1876 that his father said to him. To my deep mortification, my father once said to me, "You care for nothing but shooting, dogs, and rat catching, and you will be a disgrace to yourself and all your family." Well, the group gathered here tonight has done a lot of not necessarily in the rodent family, but it's the weasel family. It's been a long time in the last almost 50 years now of weasel catching or ferret catching. And I think they've done a fantastic job of how this all happened. And tonight we're going to hear a lot of what's happened just in the last year and what may be going to happen in the future, dealing with genetic engineering, so to speak. But tonight I wanted to bring you up at least. Most of you know most of the history of all this. But as you know, this all got exciting in 1981 when John and Lucille Hogg noticed one night their dog was doing a lot of barking. They didn't pay much attention to it, but when John went out the next day, the dog had this funny animal he was dragging around the yard. It was already dead and may have been dead when the dog started, Shep. But he kind of threw it over in the pasture, but Lucille got curious, went and scooped it out. Interesting looking little animal with black tail, black feet and all. So she carried it into Meeteetse. And here the story changes from a little bit where it's mostly been. She took it to the vet knew what it was, and they called in, and all hell broke loose. Sierra Club and everybody else was here because we'd found an animal that was just getting ready to be listed as extinct. But it turns out the taxidermist didn't know what it was. But he knew there was a lady in town that had some biology training. And he took it to her, and she did recognize the black-footed ferret. And the only reason I know this is that I was a new member of the Board of Nature Conservancy a few years ago, and one of the fellow new members is a fellow named Paul Ulrich. And Paul, all jobs you might think is unusual, he manages the Jonah oil field in southwest Wyoming, big oil field operation. But his grandfather ran the fossil butte thing in over by Kimbra. He's the one that used to mine the limestone and had the fish in it that was all impregnated. And so Paul's got an interest in this. But Paul at that time was in Meeteetse in high school. His parents had divorced. And he didn't even have the name Ulrich. He's taken the name Ulrich back. But while I'm talking to him, he's got his phone out looking through it. I'm kind of almost irritated with him. He suddenly hands me this picture. And it's a picture of a kitchen table, red checkered cloth, newspapers out there. And on the newspaper is a dead ferret. And that was the ferret that Shep had brought in. And she's the one that actually recognized it. And so we were off and running at that point. And we do have a member in my audience now that played a special role in this. He was a young kid, high school, college age, spending most of his time in southern Wyoming looking for ferrets as part of a program. Found a few skeletons, no live ferrets. And they get this call right when they were about ready to terminate and start filling out the papers for extinction, that there was a ferret found in Meeteetse. And so, Dennie, tell them what happened after that. If you'll stand up and give them a quick review of that night. You got a minute. 1 minute, but it's a great story of Dennie was out that night with a partner, and they were looking for ferrets up on our ranch.
Here, talk, look around, and we went up. We spoke to, we went in and we started to look around. We had to figure out where the Pitchfork Ranch was. We didn't even know where that was at first. And then we went to the ranch. We talked to Jack Turnell, who was the ranch manager, and Jack was a little unsure of everything. He said, I don't know about all this, this is all new. I understand something. It's an endangered species. There was a lot of boom going on in the oil industry. They were up there doing all kinds of doing surveys where they were thumping the ground, and there was just a lot of different things going on. Anyhow, he said, okay. He says, if all you're going to do is drive around with a spotlight, that's fine. You can do that. So we did. We went out. We looked around. We did some spotlighting. We saw a couple places. We didn't see anything that we were looking for in the way of tracks or skulls or any of the diggings that are normally associated with prairie or with black-footed ferrets. But we found an area that we thought, well, okay, we'll come back here in the morning. And if you know where the Meeteetse game check station is, that little wide spot just as you come out of town, we had a little house trailer there we spent the night in. And we got up the next morning, we went to the Pitchfork, and we drove up, got up on top of we're by what they call the airport, up on the plateau there, and we started looking around, and all of a sudden, I was on the right side of the vehicle. Steve Martin was my co-partner. He was a full-fledged biologist. I was up and coming. And all of a sudden, with my spotlight, I saw the eye shine that I'd been trained for 4 years to look for. This really turquoise, bright green eye shine that you just don't see in any other animal out there. Weasels come close, but nothing else shines the way a black-footed ferret does. And I saw the eye shine, and before I could say anything, the ferret ran across the road as I'm shining on it, and then it went down a burrow. And I looked at Steve, and Steve looked at me, and I said, what did you see? And he said, black tip tail, black legs. I said, mask, had the saddle. And we both looked at each other and went, ferret! And we jumped out and ran over to the hole where the ferret went down. And this was really funny because Con Hillman, who is the only other person to ever studied ferrets before this time, he had told us that when he was in South Dakota, he would take his hat off and put it over the hole so that the ferret didn't come back up out of the hole. He just thought that the burrow was plugged at that point. So I walked over to the hole and I looked down, and just about the time I looked down, the ferret came back up and he barked. Nobody said anything about barking ferrets. And I about fell over backwards. And I just instinctively, I had a Scotch cap on, I just stuck it over the hole, and we both looked at each other and we just started to dance. It was just a fantastic moment, and I'll never forget it, and it was the start of everything to come, good and bad.
Thanks, Dave. That's good. Fish and Wildlife and Game and Fish and all, at that point, started routinely following them. At one point, I think the population was as high as 80, 84 in one survey. But over the next 4 years, this number kept going down. And so it became pretty clear there were several things going on. One is, as you all know, the prairie dogs, there's a lot of plague in our ground, and the fleas up there. So the prairie dogs cyclically get plague, and they start into epidemic of it there. And this really hits the ferret hard. But also, some people had dogs up there, and some of these ferrets got distemper. First 6 they captured actually died of distemper. So things weren't looking good about 85. And here comes another heroic couple, two veterinarians by the name of Tom Thorne and Bess Williams were working in this, and they came to the conclusion that even though the ferret had been tried to breed in captivity, they couldn't. They hadn't at that time. But they made the decision that the way things are going, they're going to be extinct up here in a couple of years. So they made the decision to let's gather up what we can. So they went out over a period of time. It was a very controversial decision. Town of Meeteetse didn't like it. Some people still grumble about it. But they captured 17 ferrets. And then the night before they left, they said, we might as well go back and look, see if there's anything else. And they caught one more ferret. And I think it was over on Allen's place, if I remember. But it had a scar on its cheek. And so they nicknamed him Scarface. And they took them all down to Siberian Canyon, where they set up their breeding setup. And the only one that bred was Scarface. He bred 7 of the females. And from that, we now have our current population of over 10,000 have been bred. We're in the 10,000s now. But as you can see from this, genetically, this is not good. Because every year goes by, the genetic diversity goes down because we're not adding new genes to the pool. And so this is really the main topic we're going to be addressing tonight, is how we can try to improve the genetic diversity in this group. And so in 2016, a group of partners, Revive and Restore, the San Diego Global Frozen Zoo, and Biogen Pets and Equine, joined with Fish and Wildlife Service to pursue the idea of cloning a black-footed ferret. And this is all possible because of one of our speakers tonight. He was only 42 at the time. He's a little older now, but he did have the insight to freeze these. And that's going to be part of the major part of our presentation. And also in 2016, we had begun doing some work on the Pitchfork with fish, game and fish. A vaccine to plague had been developed in Wisconsin. So they started first as early as 2012. They started surveying what we had, how much plague was up there. Then they did a nice double-blind study where people putting out the vaccine comes in a little red pellet, has some peanut butter in it. That's why they eat it. And they picked 4 40-acre sections in 4 different sections. And 2 of these were placebo, 2 had the vaccine. Even though they hadn't broken the code, the people working up there dispensing this and capturing prairie dogs and testing them, they knew within a year or 2 which sections had the vaccine. Because suddenly populations were growing, they weren't finding dead prairie dogs around. Whereas in the other sections, they were still seeing plague. And so then, earlier than planned in 16, they decided, let's release some. So we released, I think, 37 or 38 ferrets that first year, another 20 some odd in 17. And the counts looked good the next year when they went by. Things were kind of thriving. And then starting in about 18 or 19, we started seeing the plague come back. And the counts were going down, the prairie dog counts were down. Now, this last summer, looked good. When they went up, Andy's here, and I think did the counting, yeah. I think they found 12, I think, ferrets, and 6 of them were kits, meaning they were born there. And that's a good sign. One was a litter of 4, I think, and 1 and 1. And so things are looking up right now. It's a year-by-year thing, as you're going to hear about from the professionals that do this. And so tonight we've got a distinguished panel, and it's my pleasure right now to introduce them. Zack Walker is here with he's the non-game wildlife supervisor for Wyoming Game and Fish. Zack, come on up. And then also we have Ollie Ryder. Ollie is the Kleberg Endowed Director of Conservation Genetics at the San Diego Zoo, Wildlife Alliance, and the Frozen Zoo. And Ollie has an interesting history here. He worked at the San Diego Zoo way back in the 1980s. Feels like 1980s. And there's a quote I was given tonight by Pete Gobar that I thought was very appropriate. Is Aldo Leopold, a very respected scientist. He said the first sign of intelligent dickery is keep all the parts. Don't throw anything away. And that's what Ollie's famous for. 2 of the ferrets that had been brought in of the 18 and were sent to the breeding area who hadn't bred, as they got older, they were sent to the San Diego Zoo. They died, I think, in 1988. But when they died, Ollie was the one that made the decision to not throw them out. Don't throw out the parts. He froze them. And without that decision, we wouldn't be here tonight talking about all this. And also tonight we have with us Ryan Phelan, who is the with Revive and Restore. And you all can all come up here and take a seat. We'll go from there. All right. Okay. Very good. You're probably right. And she's going to speak to us tonight also. And then finally, from our own center, we have Robyn Bortner, who years ago showed me around the center. If you've never been, and Kimberly, you're not listening to this, but I do tell you, if you do get a chance, you have to call Kimberly. It has to be arranged ahead of time. But if you ever really want a fascinating few hours, is go by the Ferret Center and see all what they do and what they've got. It's a fabulous experience that you will always enjoy. And so with that, I'm going to turn this over to Zack Walker. And Zack, you're first up. And I got some questions for you.
So a lot of that too, you know, Lenox kind of covered some of the stuff that we've done through time since 1981. And so I'll just kind of touch a little bit on that again, and then I'll add a little bit about some of the areas that we have elsewhere in the state, such as Shirley Basin. So, you know, just a quick timeline for what we've kind of been happened is, again, in 1981, and I'll go through each part of this here in a second, was a rediscovery there at Meeteetse. And then in 86, they were, as mentioned, they were captured and moved to Siberian for captive breeding. And then in 1991, the Game and Fish and his partners moved some ferrets, reintroduced ferrets in the Shirley Basin. And I'll kind of talk about that here in a second again. And then I have the kind of the next thing, you know, ferrets were being reintroduced in the Shirley Basins, populations were slowly growing. And then in 2015, we had a statewide 10J designation, and that really paved the way for the ferrets to be released up in Meeteetse. So basically, 10J ruling is that ferrets within Wyoming, it covers the entire state, at least in our instance here, are considered candidates for listing. Correct. That's and so it relaxes some of the restrictions if private landowners have them on their property. So it made it a little bit easier to reintroduce ferrets, and that's why we were able to move forward with ferrets in Meeteetse so quickly. And then again in 2016 we had a reintroduction of Meeteetse, and then in 2018 kind of the next step that we've done is create a ferret management plan. All right, can people hear me better? And then in 2018, we've adopted a ferret management plan that was approved by our commission, and that kind of guides what we're going to be doing moving forward, such as how many populations we have to, have in the state, and what the population numbers look like. So that's kind of the next step. So now I'm going to kind of go back through and show some pictures and then talk about this a little bit more. Again, as Lenox mentioned, it all started with John Hogg and Shep the dog. I won't kind of go into that story again. And but from there, the ferrets were found. We were trying to figure out what to do, how to do it. They already were considered extinct before, so there's a big fear for, let's make sure that we do it, hopefully try to do it right and we don't lose them again. So there's probably you know, a lot of steps and a lot of concern that was going on at that time. And then I have to admit, you know, I wasn't around. I can remember hearing about black-footed ferrets being rediscovered when I was in elementary school. So I'm not in this picture if you didn't guess. So, but this is the, I believe from the notes that I had, the first Wyoming black-footed ferret working group. And these are kind of the folks behind coming up with the plans for how to move forward with this species in the state. And then in 1986, the Game and Fish, again, we moved those ferrets to Siberian Canyon. And this is actually some pictures of some of those initial breeding pens. And, you know, Tom Thorne and Beth Williams were pretty instrumental in figuring out some of this, the captive breeding for the Game and Fish. You know, I wish they were here today, but I do want to, they ended up passing away in a car accident, but we did name Siberian Canyon. It's called the Tom Thorne and Beth Williams Research Station at Siberian Canyon. So, you know, their efforts for a lot of this was remembered. And then in, as I mentioned, 1991, ferrets were introduced in Shirley Basin. So Meeteetse still had plague impacting it at the time, and Shirley Basin was one of the most intact prairie dog colonies. And they're in whitetail colony as well. So in the state of Wyoming, if you weren't aware, we have black-tailed prairie dogs and white-tailed prairie dogs. And Meeteetse is white-tailed prairie dog, and then so is the Shirley Basin site. So they were decided to move them there. And yeah, I wish I knew where exactly this picture came from. I believe it came from a newspaper, and it's just been sitting in our files like and used for almost every presentation. So I had to make sure I included it. But yeah, Shirley Basin or bust. They're going to get reintroduced there. So Shirley Basin was actually the first reintroduction site, and it's the longest running reintroduction site for the species. You know, Lenox did talk about the SPV trials. So basically, kind of jump back in just a sec to jump back to what I was just talking about. In Shirley Basin, we had a very slow population growth. Populations, you know, crested up over the course of about 20 years, 30 years, and then we had a plague, we believe, plague or drought. We kind of think plague. Populations went from a little over 100 that we knew about down to in the 20s. So it was a kind of a big drop, but since then it seems like they have kind of stabilized back. And then I'll talk about some exciting news in Shirley Basin in here in just a second. But again, we did some SPV trials up in Meeteetse on the Pitchfork, and that helped also pave the way for reintroduction up there, in that we had prairie dog numbers. We knew how many were out there. We knew populations were pretty good at that site. We had willing landowner cooperation, and so we decided to reintroduce them there as well. We also do plague control. So in addition to the SPV, we work with wildlife services. And something I'm going to throw out there too, at least the Wyoming management of ferrets is a very cooperative kind of aspect to it. So we work with wildlife services. We have a working group kind of with the Department of Ag, Weed and Pest, you know, all kinds of partners. And none of this could happen if we didn't also have the cooperation with the landowners, and who were really kind of acknowledges kind of one of the driving conservation folks here in the state is the landowners we work with. So a big thank you to Lenox and to the hogs. So. And then it was the release in 2016. So I got some photos of that. So this is folks participating in that release. And I had to throw in there a picture of some of the releases. There's Lenox kind of walking with one of the ferrets getting ready to let it out. And then they were also released on the Hogs Ranch, the Lazy BV. And so that's kind of going back to where Shep first found, you know, that first ferret. One of the things I do want to point out too, since we've been up to since then, is the in Shirley Basin, we kind of pilot studied this up in Meeteetse, but then we did a kind of a more landscape effort to use scent dogs. We worked with working dogs for conservation out of Montana, and we surveyed large swaths of Shirley Basin because it's so large. It's just really not feasible to search for ferrets. Spend 3 nights spotlighting a ferret across the entire Shirley Basin. So we tried to clear out some large swaths using scent dogs. And Andy, how many new populations? 2, 3? We'll say 3 new areas that we didn't know that we had, ferrets currently. So it seems like there's probably more ferrets in Shirley Basin than what we originally kind of thought with some of our standard surveys. And then poor Andy this year is like probably going on 8 weeks of sleeplessness, trying to survey all those areas for ferrets that the dogs detected them. Basically, when a dog detects it, we set up a camera, and if ferrets are detected with the camera, we go back and do surveys there, and then you can see some of the interesting things we got on some of these trail cams set up where we had ferret detections by the scent dogs. So anyway, that's kind of what we've been up to, doing standard surveys in Meeteetse and Shirley Basin every year, spotlighting surveys, and we'll continue, monitoring all our populations and trying to keep with plague control in Meeteetse. So I guess, well, I think that was my. Oh, on the bottom left, back. Top right's a white-tailed prairie dog. Yeah. Yeah. So Lenox was asking what the difference is between whitetail and blacktail and kind of their habits. So the easiest thing to identify them is one has a blacktail and one has a whitetail. By their yeah. I was just being silly. But the whitetail prairie dogs are more sparse. Basically, they don't live in quite as dense of colonies. They usually have more dispersed colonies, and they browse. Because they're more dispersed, their browsing isn't as concentrated. So a lot of times, if you walk through the sagebrush and you come across a whitetail prairie dog colony, there's a lot less noticeable about it. So when you talk about ferret recovery, you have to have larger whitetail colonies than blacktail because they are more dispersed. So you have to have the larger acreages. The blacktails, what they clip, and they live in higher densities, so you get a lot more of them in one area, and then they clip the vegetation, so they kind of mow it down, kind of like a lawn. And so usually a lot of the ranchers are more opposed to some of the blacktail prairie dogs because they are so much more density and they're kind of clipping the vegetation, whereas with whitetail prairie dogs, they're usually not as concerning to some of the landowners because they're more spread out, sparse, and not as noticeable or effect on the landscape. So.
When we, reason I asked the last point, when we started to first introduce them back in 16, I talked to a number of my neighbors, and they weren't very excited about this. And a lot of people, all they think of is prairie dogs, lack of pasture grass for your cattle. So a lot of ranchers just don't want them on the property. A lot of ranchers don't like the feds in the state coming on their property. So there is some resistance trying to get landowners to do this. And I think Friends of the Ferrets and this group that's here tonight from the center and all have done a lot to go to different states. New Mexico, Colorado, Wyoming, Montana, all have ferrets. But we're trying to increase the number of large landowners that will allow the ferrets to be brought in to there if the environment's right for them. So that's one of the big difficulties we have in trying to get more ferret populations out and around. there are certain numbers that we've picked that if it gets to a certain level, we can take them off the endangered list. And we've used that somewhat to get our political friends to get behind us on this and help us out some financially, because a lot of the Republicans out here want to get rid of the Endangered Species Act. And so anytime you can get an animal off the endangered species, everybody's happier. And that's a good goal to have, because if you can really take them off of it, that means they're probably going to thrive. And so next, Ollie, I think is up next. And Ollie is Ollie's from the San Diego Zoo. Come on up, Ollie. And he's the one I mentioned earlier. As I mentioned earlier, Ollie's did the work, all started the cloning activity. And so, Ollie, come on around and we'll get you going here.
Can you hear me okay? No. How about now? I think that. Yes. Can you hear me okay? Okay, good. All right, well, it's a real pleasure to be here and an honor to talk, and I appreciate the kind words that have been said. I think they're a little bit exaggerated, actually, but it is true that I was 42 when the sample came in. And but it was an exciting time for us, and it continues. Those of us that were involved in the frozen zoo. And so it's a term that was chosen because it would probably say something to somebody about what it was about, help you begin to talk about it. And I'll tell you a little more about it, but this is sort of what it looks like. It's these big stainless steel tanks that are actually like thermoses, you know, with vacuum thermoses, you know, and so the liquid nitrogen is in the bottom of them, and the vapor keeps the temperature at minus 297 Fahrenheit. That's 75 degrees Kelvin, 75 degrees above absolute zero, and life almost stops there. So cells, living cells can be put in suspended animation at that temperature. And the discovery about the possibility of doing that was really only made like 70 years ago. But then people started to learn that they could freeze cells and thaw them, and if they protected them with cryoprotectants. But there was a method that you could freeze cells and thaw them, and they would resume their normal function. So that was undertaken by my mentor, Kurt Benerschka, who, because he wanted to study chromosomes of animals. Chromosomes, he's a physician. Chromosomes are important for understanding birth defects and conditions like Down syndrome, trisomy 21, and other abnormalities that, as a pathologist, he encountered in children and adults. So anyway, there was a reason for establishing these cells, but at the time we knew it was inculcated into us that, you know, science was going to move forward, and we knew that there would be things that we weren't really thinking we couldn't imagine that would be possible to do. So some people wonder, well, how do you really establish a cell culture? Well, you take a little piece of tissue, biopsy. Some of you are familiar with biopsies if you've been to the dermatologist. Take a little piece of skin. You can cut it up into small pieces, and you can basically kind of like make a smoothie out of the cells so that they're in a suspension. And you put those into a flask with a medium that supports their growth. And then they sit down on the bottom of the flask and they start to divide. And you can then collect them off the bottom of the flask and freeze them. And if you use the right cryopreservative, you can keep them alive. And it's not that hard to do to keep the skin cells alive once you grow them. Not every species is easy to grow, but that's another story. So we've, at San Diego, we've been doing this since 1975. So we have the biggest collection of its kind in the world. And it represents both the cells like skin cells, and it also represents reproductive cells like semen, like sperm. And we can get the sample of the skin biopsy can be gotten from a living animal, or it can be gotten from an animal after it's died. And similarly with the reproductive tissues, sperm can be gotten from donor males when they're alive, or from after they're dead by collecting sperm from the epididymis. This epididymis is a tube that transports the sperm from the testicle. And the sperm sit there, so it can be collected and frozen. So that was a rationale, and we started collecting cells and learning and trying to focus on species that were little studied or not known, including especially endangered species. Now, we realize that, this is now an unprecedented resource. Because we started collecting so long ago, we have material that is going to be extremely valuable in the future. And it's possible now to do things like clone animals from cells that are in the frozen zoo, or to make pluripotent stem cells that can make any cell in the body from cells in the frozen zoo. And these are cells that can actually have babies because they can be directed biochemically to produce eggs and sperm. This has been done only in the mouse, but that's just the one that's been worked on the hardest. So this makes really new possibilities. We understand that this is this a new world of conservation? As some people are concerned, are we going to not worry about saving habitats if we can do this kind of thing? We don't see any evidence that people are less interested in saving habitats because this technology is available. We rather see that there's a chance of saving more species. So the frozen zoo started in 1975, as I said. This is a picture from 1976 of Dr. Bernershka and Arlene Kumamoto, showing one of the earliest tanks we had. And this drawing is from a natural history book. I don't know if I have no idea who the ferret it was that is depicted here. But the idea that I don't have a picture of Willa, this female. And I don't know, maybe the Wyoming Department of Game and Fish has individual IDs, mugshots of all the ferrets. I'm not sure. But it's very interesting to me that the discussion about Shep and the it's just visceral image of the kitchen table with the tablecloth on it and the newspapers laid out, and a wonderment of a dead animal that's actually means that an extinct species was here. And so how many more are there? And the excitement of these early days is still palpable, just still a wonderment. Well, we froze a couple of black-footed ferrets from those early days. I don't have time to tell you the whole story, but one of them, when we went to try to say, well, who is this? It's a male. And we have these IDs for it. And the stud bookkeeper said, I don't have a record of this animal. And he had to dig deep to find out who this animal actually was. And it turned out that it was given stud book number 2. The animal that Shep brought in was stud book number 1. And this male was stud book number 2. And his cells still haven't been used for cloning. So I just want to say that I was keen to see that we could put as much as possible into the frozen zoo. And I was active in the beginning of the Society for Conservation Biology. And the first annual meeting was held in Bozeman in 1987. And that was about the time that these ferrets were being brought into captivity by Tom Thorne. And Tom Thorne attended that meeting. But before I tell you about Tom Thorne, I want to say that I also attended the meetings of the of the American Society of Mammalogists, and that group heard presentations from biologists who work for the Wyoming Department of Game and Fish and the U.S. Fish and Wildlife Service about their efforts to find out how many black-footed ferrets were there and what their range was. And they showed, I mean, really not great pictures, you know. I mean, these are pictures that are taken, you know, with good equipment and everything. But, you know, look out here at this field, and there's two little dots shining out there, and the audience would erupt into applause because it meant that they had found this, talk about that specific eye shine, that they were identifying ferrets. And they were trying to figure out whether they were males and females and how many there were, and it was electrifying to us. So when with this background and the fact that there was international attention from the International Union for the Conservation of Nature and other conservation organizations about the conservation of the ferrets that focused attention on Wyoming and Meeteetse and the Fish and Wildlife Service. So at this first meeting of the Society for Conservation Biology, Tom Thorne was there and gave a talk about black-footed ferrets. And at the banquet, I sat down next to him, and there were bottles of wine all over the place. And we drank a bottle of wine, and I just hammered on him, you should see that these samples get sent to us, because we will freeze them, and we'll be able to study the chromosome number. At that time, the chromosome number of black-footed ferrets was unknown. It's different from domestic ferrets, by the way. And so we were able to, about 6 months later, a box arrived at the lab addressed to me, and it had samples from a female who died, who had reproduced, but her offspring didn't reproduce. So there was no genetic representation in a living population from this female named Willa. And the Frozen Zoo team established a cell culture and froze the cells in early of 1988. And they sat there for years and years and years before our next speaker came onto the picture. But I want to celebrate the excitement that comes, that's about nature, that this story entails, that there's an animal, a weasel-like animal that's difficult to see, that occupies a part of the great prairies of the plains of this country, and that it was almost went extinct. It was thought to have gone extinct, and then it was found, a surviving population was found near here in Meeteetse, and people went to work on it, and their efforts made it possible for the animals to be counted, to be trapped, to be brought into captivity, and fortunately to breed them, and fortunately to save some skin biopsies. Here's a picture of Dean Biggins, and there was a picture of Tom Thorne there. These folks are heroes for their work, and I just met Dennie, and he's a hero too. And I think I'm just so pleased to be here, kind of at this epicenter of this wonderful story. And it's opening. It has ways to go. As Lenox said, the gene pool of the species is really shrinking because it's a closed population. All black-footed ferrets today, except one, are derived from only 7 individuals. And that's after 15 generations or more. And so they are close kin, let's just say. And so there's really possibilities in the future because these cells were banked and because the people at the Smithsonian have banked some semen. And that possibility led up to the remarkable story that really was captured the heart of the conservation news for quite some time, about the cloning of a black-footed ferret. And I'll just close by saying, you know, are there other endangered species that you could use this technology for? And I would say, with my experience, definitely. And in one point in case is a Przewalski's horse, the true wild horse, 66 chromosomes. All domestic horses have 64. Went extinct in Mongolia in the 1970s. And from a cell line that was banked in 1980, over 2 years ago, a Przewalski's horse was cloned and was named Kurt in honor of Kurt Benerschke, the founder of the Frozen Zoo. And that was due to the efforts of Viagen and Revive and Restore and our Frozen Zoo. So we have a number of conservation projects where the future of a species literally hangs on the being able to develop this technology. It's not an adjunct. It's not a chromium plating program. It's absolutely crucial to prevent extinction of some species. And one of those is the northern white rhinoceros, where we're using stem cell technology. And assisted reproductive technologies, as they advance and as cellular technology advances, this offers hope. And the gene pool of this unique form of rhino is all in the Frozen Zoo. It's no place else in the world, although there's 2 living animals. So I just want to thank you all again, and it's a real privilege to be here. And I want to just kind of say thanks to everybody, and I'm looking forward to hearing from Ryan Phelan now from Revive and Restore.
I mentioned to you, our next speaker is Ryan Phelan, who is the executive director and co-founder of Revive and Restore. And I'm looking forward to follow up from last year.
Thank you all. It's a pleasure to be here and an honor. I'll say that. Amazing people. Let me get my clicker here. One second. Corey? Is the wrong one. Can you hear me there? Yeah. Anyway, while he's getting me a clicker, somebody has a clicker. All right. Well, I'm going to stall for time for 1 minute. That painting on the right-hand side is by one of my dear friends, an incredible artist named Isabella Kirkland. And it's poignant to today's talk because that painting features 64 species that are all extinct and have all gone extinct in the last few hundred years. I guess I could just use... And I'm the co-founder and executive director of Revive and Restore. And our mission is to help stop species from crossing this line into extinction through what we call genetic rescue. And Oliver has hinted at that term already. Now more than ever, with the changes that are happening with climate change, with habitat loss, and increasingly with wildlife diseases, we need to stop species from crossing this line. And with the new technologies of biotechnology, we know that they can play an integral part in helping species survive and flourish. So our organization is building the capacity for these tools to be integrated in the existing conservation toolbox. So think of it as complementary technology. And fundamental to all of genetic rescue is the ability to genome sequence and cryopreserve living tissue. Those are the fundamental building blocks. And with that, with new technologies on the horizon, we can actually do genome engineering, the farthest, most impactful, and potentially controversial technology. And along the way, advanced reproductive technologies play a really important part. And the Black-footed Ferret is a great example of the use of this technology. Now, I'm going to introduce you here to Elizabeth Ann. Oh, it didn't play, Corey, but, oh, I have to do one click. So what happened was Elizabeth Ann took the world by a storm. And this is a New York Times article on Elizabeth Ann shortly after she was born. The media went crazy with the birth of this endangered animal. And I have to tell you, Pete Gober and others at Fish and Wildlife Service were a little concerned. How controversial was this going to be? But it got picked up by all the media in the world. 1.6 million people saw impressions of Elizabeth Ann, and it went down to schools and children. It took the world by storm. And with that coverage, it was something like 97% either positive or neutral. There was very, very little controversy. And I think it's because this particular species is so incredibly adorable, but mostly it's because the fact that it was once or almost twice thought to be extinct and has been brought back through the dutiful effort of so many people. So I'm going to go back and tell my side of the story, which is that we've mentioned a lot of the cast of characters here who've been involved in this, but it all really started at the National Black-footed Ferret Center. And I got a call one day saying, Ryan, we've heard about your organization. We hear you're working in this incredibly innovative technology. What could you do for the black-footed ferret? We've got some challenges. So I flew out to Fort Collins, and you can see Oliver there and Pete and you know, the cast of characters, Robyn, who's our next speaker. And it was an amazing meeting, because at that meeting, I heard for the first time about what happened with the black-footed ferret, how incredibly distributed they were from Canada to Mexico, all across the American prairie, and how with the changes in farming and agriculture, they were wiped out, and with plague, and that this one population was in Meeteetse when they were brought in by Fish and Wildlife Service, and that those 18 different individuals came into captivity. And through this incredible effort of just dedicated people, I learned that they were being released back into the wild, but that they had challenges, and that maybe our organization could be of some help. And that those challenges were not only because they were all so closely related, but also with sylvatic plague. And I'm going to get to that too. What I learned at that meeting is what conservationists call the extinction vortex, which is what happens when you have a small population of a species and it continues to inbreed. It creates lower genetic diversity, it creates lower fertility rates, it can create a susceptibility to disease as well. And so with genetic rescue, our hope and our plan here is to reverse that extinction vortex and bring in new genetic variation. Now, other organizations with other species have done this work by transplanting individuals or bringing in subspecies or closely related species from one geographic region to another. But we're actually talking about doing it with genes. And where were we going to get the genes? That was going to be at the San Diego Frozen Zoo. So it was at that meeting with Oliver that I learned that there were these 2 frozen cell lines. And no one had really planned to clone them yet. And because, as Oliver said, when they went in the zoo back in 1980, cloning hadn't even started. It wasn't until 1996, with the breakthrough of Dolly the sheep, that it was even possible to clone. So it was 8 years after they were frozen. Now, fast track decades further, cloning has gone mainstream. You may not know it, but it is standard practice in the cattle industry, in livestock, with horses, in the equestrian world, now with pets, cats, dogs. But it had rarely been used to even apply it for conservation other than as a model organism. But we really wanted to look at that cell line that was in that frozen zoo and say, what genetic variation could it bring back? So Revive and Restore, which was just a small nonprofit based in California, said, we're going to raise the money. We're going to pay to sequence those 2 cell lines and find out how much genetic variation it actually has. Willa and Stedbook number 1 actually have 3 times more genetic variation than any living ferret. So if we could go back, we thought 40 years, we could introduce all that new genetic variation and create a more robust population. So we also knew that we didn't want to endanger any black-footed ferret. And so after years of working and getting a permit with Pete Gober and everyone else and having whole genomic working groups about the pros and cons and the ethics and thinking through all the challenges of this, we fastened on the idea that we could use a domestic ferret as a surrogate. Domestic ferrets are used as model organisms to study a lot of human disease, especially lung diseases. And we knew that they were capable of easily being handled for these kinds of procedures. So basically, we took that skin cell that was in the frozen zoo of Willa, now 41 years ago, and we used the eggs of the domestic ferret. You remove the nucleus of the domestic ferret egg, and you implant the genetic code of Willa. The embryo grows up and cell divides, becomes a blastocyst. Viagen, the leading cloning company, Revive and Restore, hired to actually implement this technology. And miraculously, a clone of Willa was born. Now, we wondered, especially the vets like Robyn at the Ferret Center, was it actually a black-footed ferret? When it was so little, it didn't have all that coloring and differentiation. But it turns out within 21 days, you start to see some of the distinctive characteristics, and everybody there at the Ferret Center was marking every measurement and dedicated time to make sure that Elizabeth Ann was maturing normally, and she did. And nature was more important than nurture. She became as aggressive and as shy and behaved just like any other black-footed ferret. So unfortunately, Elizabeth Ann was not able to breed this year as we had hoped. And she has had to have a hysterectomy for unfortunate reasons. But we have more genetic twins of Elizabeth Ann in process, and they will be on their way in 2023. And we hope that that studbook number 1, who's a male, will also be part of the 2023 research that's underway, because that would be bringing back 2 new founders. So instead of 7, everybody being related to just 7, these 2 would bring in a great new opportunity. But the challenge, again, remains with plague, and cloning will actually allow for some of this innovation to go forward. So you guys all know about the threat of plague that is here in the American prairie. You know it's all over the West Coast, and you know that it's spread by a native flea. So good luck getting rid of plague. There's really no easy way to think about it. It's here to stay, unfortunately. And luckily, the research that has been done here with the black-footed ferret has allowed for an injectable vaccine to progress. But that injectable vaccine really only works well when they're captive bred. It doesn't protect the wild-born ferrets. And what that means is that this population will never be sustainable without human intervention unless they can get this kind of immunity. So Revive and Restore is actually very excited to say, this is the first time we've said it, that we've just received a U.S. Fish and Wildlife Service Recovery Challenge Grant. And part of that grant fund is actually helping us do the foundational research to develop what we are referring to as a genetic vaccine. This vaccine would actually mimic the same active ingredient in the existing vaccine, but it would be expressed when a wild-born ferret was 3 to 6 months old. So it's time sensitive. It's also place specific into the muscle tissue, just like when you're getting your COVID-19 vaccines, if you've done that before. And it would actually protect ongoing immunity for plague. Now, this is a radical idea. It's foundational research. No one has ever done this in a wild species before. However, this kind of work does happen in agriculture, and it happens with livestock, and this work is being done at lots of different levels. And one that I've been particularly focused on and tracking on since I started this organization has to do with a plant. And it's a very good model. What's missing from this beautiful landscape in the Smoky Mountains is the majestic tree that lived there for centuries. And many of you may know about the American chestnut tree. Who here has heard of the American chestnut tree? Okay, so big deal on the East Coast, but well known worldwide. It was the most robust tree and the single most successful tree across the eastern deciduous forest. For centuries, that tree poured down prolific nuts that fed humans and animals alike. That lumber of the American chestnut tree was used to create some of the most beautiful instruments in America and fine furniture. And it was decimated by an invasive pathogen. And by the way, in 1800s, there were 4 billion American chestnut trees in that forest. 4 billion. And they were totally wiped out. By 1950, there were none left, just their roots. And every year, some of them sprout up, but within 15 years, they succumb to the chestnut blight. Now, that invasive pathogen was brought in through the agricultural trade, not on purpose, but once it came, it's here to stay. And for 4 decades, scientists have been working to figure out how to breed blight-resistant chestnut trees without success until scientists at the State University of New York determined that with one transgene from wheat, they can make the American chestnut tree virtually resistant to the pathogen. It actually coexists with it. And it doesn't change anything about the American chestnut tree, just like we don't want to change anything about the black-footed ferret. We just want to add immunity. So these technologies, and by the way, I should mention that, maybe if I click on that, it'll play. No. Sorry. Thank you, Corey. Anyway, these American chestnut trees, which I have seen at the State University of New York, are in the process of going through regulatory approval with the USDA. They're the friends of the American chestnut tree that are really helping propel this idea forward. They will be restored into the wild in the next few years, I'm sure of it. So the bottom line is that with these new technologies, for some reason, maybe I'll just try again. I'll just pass that one. Both with the American chestnut tree and with the black-footed ferret, these are species that in some ways have been called keystone or umbrella species. They play such an important part of the landscape. And purely maintaining habitat is not going to be a solution. You have to use all the tools in the toolbox. Humans have taken their toll on wildlife. Go back 10,000 years. It was a very different planet with robust wildlife and species with great bioabundance. And I hope that we're right here on this timeline where with using new technologies and with new commitments to biodiversity, we can build a better future for all. And lastly, let black-footed ferrets forever.
Well, that was a very exhilarating talk. I think it got all of us kind of waiting to the next time we have a meeting and see where we are. That was wonderful. It really was. Our final speaker tonight, and this is from our own center, Robyn Bortner has been at the center how long now? Over 10 years. She's been at the center and helped give my first tour of the place. And so it's a pleasure to have you here. And she's going to tell us a little bit more about Elizabeth Ann and the center and what they're doing.
We'll be fine. We'll just point the mic down like a such. Okay. Thank you for having me here tonight. Lennox, I'm relying on your prompts. So what's your first question for me? Sure. So I'll talk a little bit about Elizabeth Ann. Let's see. So you guys have heard most of the story at this point. But I was actually there the day that Elizabeth Ann was born. I am not the veterinarian who delivered her. You saw a photo earlier of Dr. Della Garel. She's who actually delivered Elizabeth Ann. She was born via cesarean section. She had a sibling that did not survive. And I was in the room when the vets were reviving her. So this is all very unprecedented. This is the first cloned North American species, endangered species. And it's really the first time that we've tried this in black-footed ferrets with the domestic farragut surrogate. And so a lot of new horizons. We weren't really sure what would happen or how the process would go. And her mother, this is pictured here, is not the domestic ferret that gave birth to her, but the domestic ferret that raised her. Her mother, her natal mother, still survives, but she, recovering from the cesarean section, wasn't nursing well, so we actually fostered her to the mother that you see photographed here. I walked into the room. The vets were performing the cesarean section. They removed Elizabeth Ann's sibling that did not survive, and they were attempting to resuscitate her. And they had her out on the table. And Elizabeth Ann was also there, and the vets had actually, at that point, they had delivered her. She had not resuscitated immediately upon delivery, and they had her off to the side while they were working on her sibling. And I walked into the room, and as I walked by, Elizabeth Ann, just a 0-day-old little pinky kit, gasped in front of me. And I was like, you guys, this one's breathing. And they leapt into action. We resuscitated Elizabeth Ann, and she grew into what you see is the happy and healthy black-footed ferret that she is today. But it was pretty groundbreaking to be there for that experience. So she was born to a domestic ferret. We fostered her to the animal that you see pictured here, who raised her. She had some domestic ferret siblings. Born in December is outside of the natural breeding period for black-footed ferrets, because that's when we had the resources to start on this project. And we wanted some extra time in the year to concentrate on this as well. During the regular black-footed ferret season, we're very busy with black-footed ferret natural births. So we needed the domestic ferrets to raise her, and we needed siblings for her to be with. If she didn't have this foster litter of domestic ferrets, she might not be here today, because it's very difficult to raise a black-footed ferret of that age at just 0 days old and nurse them without a mother. So we're very fortunate to have those resources available. And her surrogate mother raised her, and she did great with her domestic siblings. And I think up until just before her eyes opened, black-footed ferrets are handleable up until about maybe a week to 10 days after their eyes open. And then all of a sudden they realize that you are not a ferret, and perhaps they should be afraid of you and bite you. and they will. They bite a lot. So black-footed ferrets stay very wild. They're very defensive. They're very aware of humans. Elizabeth Ann was very timid up until her eyes opened, and was often on the bottom of the heap with her siblings. And we monitored her really closely. And my vet, Della, we were always worried that she was being outcompeted by her domestic ferret siblings until the day that her eyes opened, and that it was like a whole new ball game. I walked in there, and all of a sudden she was wrestling her domestic ferret surrogate siblings. She was the first one on top for food, and then shortly after that she started becoming very defensive and wary. As Ryan mentioned earlier, nature quickly overtook nurture. She became very wary of her human keepers. All the other domestic ferrets would be out eating in front of you, and she would have run up the tunnel to hide from you, and she would bark at you and be defensive, and no longer really tolerated her handling, just like our normal black-footed ferrets are. So to be able to watch her through this entire development was a really exciting project to be a part of, all this groundbreaking science that all of our partners have made happen. And it was really exciting to be there. So yeah. Mm-hmm. Yeah, you can pick them up before about 40 or 50. That's when they start to bite you. Up until that point, yeah, and then they're pretty defensive, and you have to use a handling cage to work with them after that. Let's see. let me back up one. I'll just touch on the recovery implementation team real quick before I address that, Lenox. The Black-footed Ferret recovery implementation team and Black-footed ferret recovery in general is a very partner-driven project. We have over 50 partners that work in this program with us. We work cooperatively with each other. It's the main governing body of this is the executive committee, which is usually comprised of state agency directors. And underneath that, we have a series of subcommittees. And these are all partners working cooperatively with each other to further ferret recovery. And the fact that it's all together and works is really amazing with how many people that we work with. The conservation subcommittee is what we really call like our boots on the ground. This is where Zack and all of our state agency partners and these guys are coming into play. These are the release sites. These are the people out there working with animals on the ground. This genomics project actually falls under our managed care subcommittee. So the managed care is the subcommittee that governs all husbandry and care and transfer and management of black-footed ferrets that are held ex situ or outside of their natural range. So animals housed in captivity, if you will, in cages and centers and breeding centers, not ferrets in the wild, which are in situ animals. And the genomics project is actually nestled underneath the managed care subcommittee, and the genomics working group under the managed care subcommittee has actually grown probably larger than the managed care subcommittee itself. We have a number of partners working on this project to further that. But to answer your question, Lennox, we have over 50 partners working on black-footed ferret recovery. This is just a sampling of some of them, and they range from everything from outreach and education to the breeding centers that I mentioned earlier, the Association of Zoos and Aquariums, and the Species Survival Plan is our largest core group in the managed care subcommittee. They are these only 5 institutions outside of the National Black-footed Ferret Center where I work that breed black-footed ferrets cooperatively. And then just the multitude of other partners that make this happen, from outreach that supports all the other subcommittees and education to the related species, which is mostly prairie dogs. Disease is mostly focused on plague, of course, as well as other diseases that might impact black-footed ferrets. And the conservation subcommittee, which is where the real work is happening out in the field, getting those animals back into the wild where they came from. It is nice, yes. Of course, yes. So the question is, what is the overall value of black-footed ferret recovery and all the parts that go along with it? I usually approach this as black-footed ferrets are just one piece out of our prairie puzzle that you see here and all the animals that go with it. So the way I usually approach this is, if black-footed ferrets haven't made it out on the landscape and they've become extirpated for some reason, it's because something out in that landscape is broken. And we don't always know what the value of that is. There could be amazing developments for science or medicine or other applications that could benefit the human population as well as all the populations of animals that you see pictured here, that we don't really understand once it's broken what it is that we're missing. So if we can repair the prairie so that black-footed ferrets can exist there again, then we're repairing it for everyone. We're putting a piece back in the puzzle, and it's putting the prairie ecosystem back together the way that it used to be to support all these other species that fall under this umbrella of conservation that can be extended to black-footed ferrets that can then go to all the other species, because the prairie dogs really are the drivers of this ecosystem. But by using black-footed ferret recovery, an endangered species, we can make sure that we extend protections to all these animals. And if we put this puzzle back together and we restore the prairie, we could be doing a lot of good for ourselves, and we just don't know what it is yet. So. You're welcome.
You all have been a very attentive audience, so it's your turn now. We're open to questions, and any questions you've got and who you want to direct it to. Yeah, if you could. Oh, you'd probably just have you come up here. Yeah. And you've got the mic for the audience. Any questions from anybody? Comments? Yes. He's going to hand you this.
In the 80s and the 90s, it was illegal, and I don't know if this was in certain states, but it was illegal to have a domestic ferret. Why was that? And what does, I mean, I know somebody who had a ferret at their college dorm, and they had to hide this because this was illegal. What is the story behind all of that, and how does that involve Black-footed?
Different kind of ferrets. And nobody's had a Black-foot ferret as a pet, I know of. But there are ferrets that people have now. That are pets. Zach, do you know the different names for them? For the domestic ferrets that are pets and all versus Black-footed, they're different.
Yeah, they're different. I believe the domestic ferrets are European.
Yes.
Yeah.
You know, I think is this live? No.
Here you go, this is working.
I think I would just answer that question about domestic ferrets, or the idea of having a non-native species is the problem at any time, and because it will displace the natives. And I think that's the simple thing why both regulatory authorities as well as just common sense conservationists will say if you can avoid not, bringing in non-native species, it's a good idea.
Domestic ferrets. But the problem with them was, one, they smell because they're musky, okay? They're part of the weasel family. And two, if they can get their head through a hole, they're gone. And so most of them got away. But just to answer your question, I never heard of any limitation. I was also a game warden for a number of years. And during that time, there were no laws saying that you couldn't have domesticated ferrets. You couldn't in California.
Some states.
And usually you descented them also, and they had to have a little tattoo on the back of their ear. But California, even when you did all those procedures, would not let you have a ferret in because of the population of the poultry. They did.
Yeah.
They would get out.
So I have a question. I used Siberian ferrets in part of my research, and I was curious about why the Siberian wasn't used as the surrogate, because at least back when I was involved, it was considered to be the closest living relative to the black-footed ferret. And I'm curious, how are the Siberian ferrets doing?
Well, I don't know how they're doing, but I can tell you that early experimentation, way before Revive and Restore got involved, using them as surrogates with the Smithsonian led to some abnormal behavior. And they felt like that was just the wrong mix. And so because we knew what we were doing was already going to be innovative, we didn't want to add any other challenge. And we knew domestic ferrets were good models. So, and easy to get. Now, I'm dying to hear more questions from the audience. I know the other panelists are. You've heard a lot of stuff here tonight. Yep. Here in the front row.
I can talk loud. I know that this was controversial at first. Was there immediate trust, or can you talk about the trust between the U.S. Fish and Wildlife Service and Revive and Restore? It's a private company, and the U.S. Fish and Wildlife Service is extremely conservative in their movements. In the last 5 years, how did the trust work to arrive at a clone?
Well, I should probably ask Pete Gober to answer that. Or Pete, do you want me to take it? Go ahead. Well, I would say that when we first came out, we were kind of the West Coast liberals in the meeting room, and we were viewed with great suspect by the characters that were involved with the Smithsonian, all these state institutions. I'm not sure if that's 100% accurate, but I felt like a real outlier with people who've been working on these ferrets like Dennie for 30 plus years. And I think the one thing that we had going for it was our honesty that we were doing this to help conservation and that we were a not-for-profit organization. There was nobody in it for the money. And we were going to be in it for the long term. And I probably got asked that by you, Robyn, and others, is Revive and Restore going to stick with this? And so I think partly it was the tenacity with our team that we were going to be in it for thick and thin, and that we were really going to work with all the partners and share the responsibility and be part of a team. And so I think that has built a really amazing level of trust with everybody, and has actually allowed us to even secure some Fish and Wildlife Service funding for this innovative research. But everything else that we did, the cloning was paid for by funds that Revive and Restore paid, as well as the sequencing. So, and we're in it for this next phase as well. So I hope we're earning it.
Process can be burdensome. The process can be your friend if you go through all the steps and step carefully. And it's not like we gave them a blank check. Revive and Restore is permitted. We've got our hand on the process.
Yeah, Oliver, you do that because you know Willa. So how does Elizabeth Ann, and she is Willa?
Well, that work is ongoing because we had material that was banked from the animals that we got, but not all of the founders were samples were there for DNA sequencing. So some of the ferrets we were looking back later after there was generations of breeding. What was the likelihood that individuals that lived closely in the last population were unrelated? It's likely that there was some degree of relationship. We're going to learn more about that. The sequencing work is going on, but as Ryan said, we can look now at today's ferrets and see that we know they must have lost genetic variation just from the structure of the pedigree, but we can measure it now, and we can see actually which genes are more, which genetic variants have been lost, and see that in fact that these cells that were banked in the 1980s have more variation, so we know that they can contribute to a kind of a recharging of the gene pool. Did I answer your question?
So you can't tell if they share a great grandmother.
So Elizabeth Ann is a genetic clone, meaning that she is exactly like Willa. So her genome is 100% Willa. No, she never bred, and if she did breed, the offspring never contributed to the gene pool. So what's great about it is both those two cell lines will be like new founders. Yes, in the far back.
So I'd like to know, when you introduce this genetic immunity, how do we know, like in the case of the chestnut tree, how do we know that the chestnut tree now doesn't have this sort of super immunity, and when you replant it on the East Coast, it's going to like not die, and it's going to displace a lot of other trees that should be growing there? So, I mean, what's the safeguard on that?
Yeah.
I mean, when you introduce this, genetic immunity, like a genetic vaccine, I mean, how do you know you're not producing something,
I hear where you're going, and it is obviously something that has to be studied. And part of what we have built into the work that we're doing with U.S. Fish and Wildlife Service is those kinds of safeguards that, we're looking at, there's no one going to even introduce these clones themselves into the landscape because, number one, they're way too valuable in and of themselves, but we want to see successful generations to make sure there isn't something aberrant that's going on. And the same with the American chestnut tree. They've gone through generations. They've studied the trees over time, and they've done very, very extensive research that they had to do for the EPA, the USDA, and some other major government body. I forget which one, but they are absolutely studying that. So, all of these things, it's just like with human agriculture and the food industry, you try things, you monitor it, and you rein it back if you can, if you have a problem. Other questions? There was somebody right up front. Yeah.
There was a lot of mention about the distinctiveness of the eye shine of the ferrets. Has anybody looked for, do you know if that's due to, do they have kind of a structure to their eye that allows an increase of photons that come into the eye?
Yeah, I don't know if anybody else wants to correct me on this, but really there's a coating on the back of the eye, the tapetum lucidum, which reflects light, and the certain wavelength that reflects back actually causes the color of the eye to shine a certain way. Another thing, like for example, when I'm out surveying, I always typically have I have problems with pronghorn. They have a very close, it's not that brilliant emerald green, but there's kind of a greenish color, and they'll be laying on the ground, and I can't tell you how many pronghorn I've followed because they have the the thing on the back of the eye that reflects. So yeah, almost like the way we can identify everything out there is by the color of the eye shine. So there's a few things that are very similar, like I believe the swift fox is kind of more of like an amber color, rabbits are red, so it just, when you hit it, you get a different color back, and you can identify kind of what you're seeing based on the color of the eye shine. Yeah.
Hi, I guess my question is maybe mostly for Mr. Walker. I hear a lot about the predator-prey relationship between ferrets and prairie dogs, but obviously 500 years ago when there were more of them, they would have had ecological relationships with a lot of other species. I'm wondering if any of those relationships are starting to reestablish to the point where you have to factor them into your work, or is that not really an issue?
Yeah, right now I wouldn't say that that's an issue. They're pretty much still a prairie dog obligate. So about 99% of their diet, they might occasionally take another rodent, but really primarily what they're feeding on is prairie dogs out there, and they usually eat about one every 3 days. Right now we don't have a lot of evidence of, having to worry about different prey items. We focus on the prairie dogs.
So, over here, I was just curious with regards to the frozen zoo, just how, what's the oldest, extinct animal, material that you have that could potentially be used to, in a way, resurrect the species, or just help preserve it that way? Just curious.
Well, thanks for the question. I really have to say, the truthful answer is I don't know. I know that we have living material from 3 extinct species in the frozen zoo, and I'm not counting the ferret. One of them, one of them is a frog, and growing frog cells is particularly difficult. And we've been able to establish and freeze more frog cell lines than anybody, but we still don't know how to do it well. It takes a long, long time to grow the cells, so it's not optimal. But we have there was a frog that was rescued from a epidemic of fungal disease that caused the extinction of a species, and there was a rescue effort to bring these frogs into managed care and breed them, but they weren't successful at breeding them. And then the last 2 individuals were sent to us, and we couldn't, we took part of the tissue and froze it in a way that we know or we expect keeps the cells alive. And the other part of it, we tried to grow, and we just couldn't get the cells to grow. But we have something that maybe in the future we'll be able to figure out how to grow it when we learn more about how to grow amphibian cells. Another one is a beautiful animal. It's a tree snail that's native to Hawaii, that evolved in Hawaii. There was many species of these tree snails. They live on trees, don't move very far. They have an incredible life history. They live to be 15, 20 years old. They don't sexually mature till they're 7 or 8. They are snails and they live on the trees, but they don't eat the trees. They eat the fungi that grows on the leaves. They're actually cleaning the leaves for the tree. And there's a Hawaii Endangered Extinction Prevention Program that said, like, there's only one individual of this snail left. Can we send it to you when it dies? And we said yes, and it's the same kind of thing. We've never grown snail cells, and not a lot of other people have either. And so we froze it, and we don't know. There's one species that went extinct in the late 1990s, early 2000s. It's called the po'ouli. It was discovered in 1978, and it was a very small population. And there was discussion, should we bring some into captivity? You know, how many are there? It's in some ways eerily reminiscent of the black-footed ferret situation. And by the time they decided, yes, we're going to bring them into captivity, they could only capture one. And it died, and we got tissue from it and have saved its cells. So we have the cells of the po'ouli. The team recently banked cells from the vaquita, which is the world's most endangered cetacean, member of the whale family. It's a small porpoise that's caught in the middle of a narco-trafficking and wildlife trafficking nightmare. And it gets drowned in nets that are because of illegal fishing activity. And it's thought now there's 10 or less of these animals, and there was an attempt to try to capture them and bring them into a kind of an oceanarium kind of situation. But the animals, when they were captured, became so stressed that one of them died. And so the operation was stopped. But we have cells from 2 female vaquita, you know, that species is possible to go extinct. And a number of birds that are on the road to extinction in Hawaii because of avian malaria. So extinction for a lot of people is kind of a vague concept. It was for me, you know, dinosaurs, well, they're extinct. But for us in the frozen zoo, it's actually something that we encounter. So I don't know if I answered your question or not.
Yeah, thank you.
So I was curious about when it comes to handling the animals. I can't remember your name. You're the one with the red hair. Robyn? So you said you were getting bitten and like scratched, and they were vicious, right? So how do you handle that? How do you handle an animal that doesn't really want to be handled?
Carefully.
Yeah. Oh, you have to think. Yeah, thanks. So I alluded to being bitten. We don't thankfully get bitten by our black-footed ferrets on a daily basis because we've gotten pretty good at how to work with them. So when they're young, you can handle them, and we do often to make sure that they're doing well, that they're healthy, that there's no problems, right? But then when they reach a certain age, you cannot. And black-footed ferrets stay very wild, and they do not tolerate physical restraint. You cannot just grab one with your hands. I've learned that the hard way. So we try to stay very hands-off. With them, we, like I said, up to a certain point you can handle them, and then after that you have to use sort of more hands-off strategies. So usually if we need to handle an animal, we use a handling cage. It's a piece of welded wire that we've welded into, I think you saw some photos of animals in the handling cages in some of the presentations. And we can do an exam in there. We can give a quick vaccine. We can give medication if we need to, but really don't put like our hands on the animals. They do not tolerate it. They'll twist out of your grasp and they'll try and bite and get away from you. So we either put them in a handling cage or we just don't. We try to just sort of monitor through, their enclosures and not get too hands-on. If you have to be any more invasive than that, you do have to do an anesthetic procedure. And we only reserve that if we absolutely need to. So there's only a few events in their life that we do that for, when they need their first vaccines, or most of our animals will get like a yearly physical just so that we can make sure they're doing okay, do a quick blood draw, do a quick health check. And outside of that, we really try to just let them do their thing, let them be wild, and try not to handle them if we need to. But we do have big leather welding gloves in reserve just in case you need to coax someone into a handling cage somewhere.
As I'm sitting here, and I certainly support this effort totally, but the question that goes through my mind relative to a lot of this kind of thing is, you can bring species back. I mean, and we'll be doing it, quite a bit in the future, but landscape is diminishing in terms of Homo sapiens. We think we have to populate the entire planet, and so I'm wondering, are you beginning to find conversations around how we'll be able to not justify, but in some way make room for these little sweethearts if we can bring them back, and we've got more and more concrete all over the planet. You get what I'm getting at?
So you get what you pay for in life. And that close? Yeah. Okay. You get what you pay for in life, and one of the good things about black-footed ferrets is there's a place to put them back now. A lot of our colleagues in the zoo community who deal with conserving tigers, for example, there's no habitat left to put them back, and human populations are growing in those areas. The highest and best use of many of the lands in the western United States will be for a long, long time, despite energy development, etc., will be grazing, livestock grazing. So there's huge areas for prairie dogs to thrive, which ferrets require, as long as you can find landowners who are okay with that. The problem with that is they're trying to get grass into their cows, and they don't want to see the grass go into the prairie dogs. But if you look at the economics of livestock grazing, you can see that they really don't make that much money on a per acre basis net from livestock grazing. So if there's a place for ferrets out there, our recovery plan calls for prairie dogs to be purposely conserved on 1/10 of 1% of the potential prairie dog habitat across the West. I'll repeat that, 1/10 of 1%, 500,000 acres. That's not a lot to ask for in a 500 million acre landscape. But you'll have to do several things in order to make a place for ferrets. One, you gotta have wildlife agencies like Zack there to manage and follow them. Two, you gotta deal with the landowners who own the property. In many cases, particularly in black-tailed prairie dog habitat, which Zack alluded to, you're going to have to pay an incentive in many cases. There are folks who will do it voluntarily, but if you're looking for large-scale conservation, you're going to have to pay in the neighborhood of $10 to $15 to $20 per acre for people to tolerate prairie dogs. And you're going to have to deal with the ancillary issues of too many prairie dogs and probably do prairie dog control at the boundary. These things are all happening across the West now with black-footed ferret recovery. Another thing you're going to have to do is plague management, because it hasn't been mentioned tonight, but for those of you who aren't aware, it's an exotic disease that's only been in this country for 120 years or so. So a lot of these efforts are to counter something that's already in a drastically altered environment. So there's a place to put them, and we've got a chance to work on them because people have conserved them over time. And if we can find other partners to work with, it takes money. It takes money for wildlife agencies to manage these species. It takes money for landowners to be tolerant of prairie dogs. It takes money to manage plague. And all of that can be accomplished, and we can conserve ferrets simply as a way, kind of a way to conserve prairie dogs, which will benefit all the other species that Robyn showed in her slide there across the West.
If there are no more questions, we've got you got one here?
Are there young people that are interested in doing what you're doing? So that we don't run out of.
I was really old. It's terrible.
No, I'm old as dirt, but I mean, the young people are excited about this. We have school kids come through our building and they see the frozen zoo and they get this. You can say to them, you know, we don't know all the things that this can be used for, and we don't have the time to work on all of this. Maybe someday, you know, you can do it. It can be, we have summer interns, and I just did an exit interview with one of them, and it was just like, it changed her life. She says, this is what I want to do. I didn't sure what I want to do. This is what I want to do. So that's you know, I mean, there's a lot of bad news out there. People say, how can you do what you're doing? You know, you're swimming against you're trying to push a rope, you're swimming against the current. You know, but the really hope is the passion of young people who want to make the world a better place and keep the wildlife that's the legacy of our country.
I have one thing. Oh, sorry. Because of the success of Elizabeth Ann, people very senior in the U.S. Fish and Wildlife Service paid attention, and as did many of the field biologists who are working in other endangered species. And one of the questions that I asked at a meeting was, what should we be biobanking today? And what should we be genetic sequencing that could better inform the management of these species? And with that, we now are conducting 2 pilots with Fish and Wildlife Service in 2 different regions, where we're working with their field biologists, especially now as these species were going to be going into hibernation, to actually obtain specimens to go into cryopreservation and to help them with genomic sequencing. And the goal is to get all 1,500 U.S. endangered species today cryopreserved and potentially genome sequencing. Right now, less than 17% of all plants and animals in the U.S. that are endangered have ever been cryopreserved to any degree, and less than 1% sequenced. And it is our legacy. And I think we owe it to these species to see what we can do to help keep them from crossing that line.
So I alluded to it when I was talking as well, but you know, I just want to throw out again that at least, you know, management of black-footed ferrets in Wyoming is really dependent on all the people who help out and all the partners. So, you know, from the landowners to the public, to federal partners, to state partners, to NGOs. I mean, if it wasn't for everybody working together, we would not be where we're at right now. And I just wanted to make sure that everybody hears that I am appreciative of everything and all the effort that everybody puts in, so.
Recognized tonight, and I just want to make sure that you all understand they're the keys to this whole program. And you've heard from one Pete, but Kimberly, I want you to stand up because Kimberly's done all this work, put it all together. Come on. But she really has done an extraordinary amount of work. Both of them have been lining up the topics, the speakers, and the meeting I think is phenomenal. I'm a retired heart surgeon. I spent 40 years going to scientific meetings. I was involved in a lot of research. This kind of session tonight is exciting as anything I saw in the last 40 years. Because it's just, its point is, it's taking something that was a real problem 40 years ago, how they recovered it on the land, and all the science that's been done recently is a phenomenal story. And I think to hear from you talking about only 17% endangered species, we obviously have a lot of work to do. And I think meetings like this can get a lot of people inspired. And so I hope we have follow-up sessions to this meeting and hear about what's going to happen with the next group of clones. And my compliments to all of you all for what you've done. I think
And
it's a
thank you to
huge
Lenox
investment.
for hosting us.
So the huge thank you to the panelists, and thank you all for coming out tonight. I know it's late on an evening night, so we really appreciate your attendance. This has been wonderful. Thank you.
Thank you for it.